So let's finish. I saved the best for last. That is the most detailed stuff. What I want to do now is, if I have, my last set of batteries. It's okay. You know, I love this green pointer, but it just is the biggest battery hog. I mean, literally, this is how many batteries I went through in the course. It's just crazy. You can go through about a pack of batteries. I'm not sure why it eats so much power. Well, on the other hand, the green laser is so much nicer than that little red laser. So if you have multiple traits, how do you look at and interpret that G matrix? That gamma matrix. And it turns out that it's hard to eyeball it and get general features, but if you look at its eigenvalues, you can actually ask quite a lot about it. So we'll be talking about, if you assume a quadratic fitness surface, what you can say about it from the gamma matrix and how you look at that. Then we'll say a little bit about non-parametric fitness estimators, because again, we talked about for a single trait, a quadratic fit is often bad, so you imagine it's probably as bad, if not worse, for multiple traits. So how do we get around that? We'll talk about pursuit, projection, regression, and thin-plate splines. Then we'll talk about another way to analyze data, which is path analysis, where I basically give you a causality structure. Trait 1 affects trait 2 affects fitness. And so it's another interesting tool to look at selection. We'll talk about latent variables, we'll talk about Morrissey's extended gradient, and we'll talk about elastic path analysis. And then we'll conclude with a quick example, and this is covered in more detail in chapter 22, which is, so far we've been focusing on fitness that's entirely based upon trait values. You could also imagine fitness is based upon some emerging or ensemble property, such as, what's the group density, right? So we'll talk about how you can incorporate group level traits, that is, traits above the individual. This is called levels of selection. It's a kind of more politically correct way to talk about group versus kin. So levels of selection basically give you a way to ask about, are there population level features that aren't features of individuals, like density, that influence fitness, and how you can estimate those. And the bottom line is you simply put them into regression and estimate them that way. Let's talk first of all about quadratic fitness surfaces. So here is our model. So again, what I'm doing is I'm asking for my data, what's the best quadratic fit predicting relative fitness of individuals, given a list of trait values? Well, and the key thing here is we want to look at this quadratic term here. And we talked a little bit about how you can interpret that quadratic term. For example, a gamma II bigger than 1 means as my trait values get larger, fitness goes up. Larger or smaller, right? As they get more extreme, fitness goes up. That's a positive curve on a selection surface. One example would be disruptive selection. If gamma II is negative, it means as I go away on either side from some value, the fitness goes down. Stabilizing selection is an example, but again, I simply could have a negatively curved fitness surface with no peak, for example. So those are kind of obvious, but as we see, looking at it character by character gives you a distorted picture of the nature of selection. You want to look at it, the entire G matrix at once. So one feature is if I have a quadratic function like this, if I take its derivative, I get that. If I set that, and so what it basically says is, if I take the average of that, the average here gives you the average slope. But the more important thing is, if I have the derivative equal to 0, that's a stationary point. It could be a maximum, it could be a global maximum, global minimum, or it could be one of these saddle points. But the key is there's only one such point in a quadratic surface, and it satisfies this relationship here. So at the point given by this, the mean fitness is here. And that's either the global maximum, the global minimum, or if I've got a saddle where one part of the curve goes up, the other part goes around. The bottom line is there's only one extreme point, no matter how many data you have, how many characters you have, in a quadratic surface. So what matters is, what's the geometry about that quadratic surface? Right? What does that local fitness look like? And it depends upon the eigenvalues of this matrix here. If all the eigenvalues are positive, and again, remember, just to remind you again, eigenvalues. So I've got gamma times E equals lambda times E. So that's a matrix. So if I have a direction, a vector, if I multiply it by the matrix, what I get out is I get out a vector pointing in the same direction. And by the same direction, if it's flipped about 0, it's still in the same direction. And then it's got a scaling on it. If the scaling is negative, it's reflected about the origin. If the scaling is bigger than 1, it increases. Less than 1, it decreases. So this is called an eigenvalue, an E-value. And these are called eigenvectors. So if all the eigenvalues of the gamma matrix are positive, we say the matrix is positive definite. And in that case, this value here corresponds to a local minimum. As you move away from it, fitness always increases. If all the eigenvalues are negative, called negative definite, then that value here is local maximum. As you move away from it, fitness declines. And we'll see where the eigenvalues come in. We do a transformation. It makes it a bit clearer to look at. And if you have some mixture, it's called a saddle point. So 0 eigenvalues correspond to directions of no curvature. Positive eigenvalues correspond to the direction of positive curvature. As you move away on either side, fitness goes up. Negative eigenvalues correspond to directions of negative curvature. As you move away, fitness goes down on either side. And so it's the eigenvalues of this matrix here that really give you an idea of what local geometry is. And let me give you an example. I'm going to give you 3 different fitness surfaces, and they all have gamma 1,1 is -2, gamma 2,2 is -1. So they all have this. Where they differ on is what the gamma 1,2 is. So here, gamma 1,2 is 0.25. And that resulting surface looks like this. Here's the topography. Here's the map. It's stabilizing selection in both dimensions. And furthermore, what you'll see is you've got sort of 2 axes. These correspond to the eigenvectors. So here is one axis, and at right angles, here's the second axis. The way to think about the axes, if I make these now the new coordinates, this thing is now symmetric. So along this direction here, the curvature is -2. Along that direction, back up. Along this direction here, notice the lines are spaced apart. The curvature isn't that extreme. The curvature is -0.9. Along this direction, those lines get closer quicker. The curvature is more extreme. So in this case, you kind of match here, right? If you look at character 1, you've got a curvature of -2, curvature of -1. When you actually look at it, you sort of see that same thing. So the eigenvalues give you the coordinates of that surface. The eigenvectors tell you how strong they are. So if you just look at this, you go, well, these things are essentially the same. -2, -1, -2 point something, -1 point something, almost the same. However, let's look at what happens if we take these exact same values and now change the gamma 1,2, that is the cross product term, from 0.25 to 1.4. That's a totally different surface. If you looked at your data and saw, oh, trait 1 is under stabilizing selection, trait 2 is under stabilizing selection. In reality, what you see is this. I have one eigenvalue that's -3. I have a second eigenvalue that's 0. So this is the direction here for the eigenvector corresponding to this first eigenvalue. I get steeper slopes than I get in here. So along that direction, I fall off quite quickly. And notice that affects both character 1 and character 2. But in this direction, I have an eigenvalue of 0. Eigenvalue 0 is a ridge. There's no curvature. Now, that ridge may have a tilt, but there's no curvature on that. So with these same data, same 2 gammas on the traits, I could either have stabilizing selection on both traits with amounts that kind of match what we expect. I could have stabilizing selection on one combination and no selection on the other, And that's simply given by tuning this value, going from 1/4 to 1.4. If I tune it even more, make it 4, I get this surface. So along this direction here, and of course that direction is given by the eigenvector associated with there, and there's a typo in here, should be a minus sign there. So along this direction, I get minus 2.5, and along this direction, I get positive curvature, so I have a saddle. In this direction, I have, this is the direction here, lambda 1 corresponding here. I have slight upward curvature. In that direction, I have large downward curvature. And the bottom line is all three of those surfaces all have the same apparent selection on the two traits. The difference is in terms of this cross product term. So if you just simply look at these diagonal elements, you can't distinguish which of these rather three different things you have. So you have to look at the full structure, and the full structure is given by the eigenvalues. Here, 2 negative eigenvalues, stabilizing selection in 2 directions. Here, 1 negative, 1 0, stabilizing selection on one, no selection on the other. Here, again, that's a typo. It should be a negative and a positive. In that case, stabilizing on one, directional on the other. So the gammas don't tell you that, but the eigenvalues of the gamma matrix do, which is why you want to look at the eigenvalues of that. That just basically said what I said before. So to get that, we need some more matrix algebra. How fun is that, huh? So a couple of terms. We have an expression called an orthonormal or unitary matrix. An orthonormal matrix, so this basically, so I write vectors as columns. This is a bunch of columns. And in an orthonormal matrix, the dot product of column I and column J satisfies this. The inner product of I on itself, that's length. So each of these vectors have length 1. You think about this vector as an arrow, and do Euclidean distance, it has length 1. So each of these columns has length 1. However, if you look at the interaction of the two columns, their inner product is 0. Vectors with inner product of 0 means they're at right angles to each other. So this is a matrix whose each column has length 1, but the columns are at right angles to each other. And when you have such a matrix, it's called unitary. It satisfies this cool property that its transpose is its inverse. And what unitary matrices do is they give you rigid rotation. So if I have 2 vectors and I multiply each vector by the same unitary matrix, it keeps the angle between them and rotates them. If I've got a non-unitary matrix, the angle may be here in one configuration. When I multiply them each by the same, the angle may change. So unitary matrices give you rigid rotations, and in a sense, this is the matrix algebra version of giving you a coordinate axis shift. The reason that's important is I can take any symmetric matrix and I can diagonalize it in this form here. I have a unitary matrix, its transpose, and this thing here is the diagonal matrix. And the cool thing here is the diagonal matrix, the elements are the eigenvalues, and the unitary matrix, first eigenvector, second eigenvector, etc. So the eigenvectors are at right angles to each other, and here are the corresponding eigenvalues. By the way, from this, you get a couple of really cool things that come right away. If lambda is an eigenvalue of A, and E1 is an eigenvalue of A, what's an eigenvalue and eigenvector of A inverse? Well, it turns out the eigenvalues are the same. Eigenvectors are the same, and the eigenvalues are simply 1 over that. So if I have eigenvalues for A, the eigenvalues for the inverse are 1 over that. What about A to the 10th power? The eigenvectors are the same as A, and the eigenvalues are simply these things to the 10th power. So a lot of interesting features about eigenvalues fall out of this decomposition. But the reason that's nice is if I multiply, pre-multiply A by A transpose, post-multiply by U, I get this diagonal matrix back out again. Now this is a critical thing. It means if I have a set of data which are correlated, when I multiply them by the appropriate transform, what happens is they become uncorrelated. So I can take a set of correlated characters and translate them to a new set of axes where they're uncorrelated. And on this scale, these cross product terms go away, and therefore there's no, these cross product terms are often called correlational selection. So it's much easier to visualize the structure if you can ask what happens on combination 1, combination 2, combination 3, combination 4, independent of all the others. And this transformation does that for you. It's called the canonical transformation, and Pat Phillips, where's Pat? There's Pat. So Pat Phillips and Steve Arnold were the ones that came up with this notion. And here's the idea. Here, right here, this is our fitness function. And what I'm doing is I'm simply letting y equal u transpose z. So we've written this expression here in terms of z. I can then replace z by u times y, because remember, the transpose is the inverse. And then that's the value of z, that's the value of z. If you manipulate that then, what you end up getting is this, u transpose gamma u. What is that? That's now a diagonal matrix. And so this complicated thing with all these cross products now collapses to this. The quadratic terms are lambda I times this new variable, and that new variable is simply given by right here. It's given by the eigenvalue. So the ith one is the ith eigenvector inner product on z. I'll give an example of that. But the nice thing here is because the cross products go away, all of a sudden this becomes easy. So let's imagine gamma 1 is positive. It means as you increase combinations of this character, fitness goes up. If gamma 1 is negative, as you increase combinations of this character, fitness goes down. If gamma 1 is 0, then there's no curvature on this character. So this character is a new combination of traits, y, a composite character, and the eigenvalue here tells you about the curvature. If it's positive, it's got a positive curvature, negative, negative curvature, 0, no curvature. So this transformation then allows us to take a complicated matrix and give it a very simple interpretation. And these axes here basically correspond to the eigenvectors. We can also do a thing which basically to tilt that plane and entirely remove this linear term. So you can imagine I've got a curved surface. That curved surface could be tilted. Those tilts are the selection gradients. I can then basically reform that so I tilt it back again, and now I'm focusing entirely on the curvature. So a lot of times we're interested in the curvature. Are there parts where I've got disruptive, stabilizing, or flat? By doing this transformation, all those nasty cross-product terms go away, and it makes it really easy to interpret what that structure looks like. And again, I gave you an example of that. So let's work through that. So if you look at the scenarios depicted on here, that scenario and that scenario, so what are we doing? What we're doing is that axis is y1, and that's the coefficient on y1. That axis is y2, that's the coefficient on y2. That axis is y1, that's the coefficient on it. That axis is y2, that's the coefficient. That axis, so that axis there is y1, that's the coefficient on y1. That axis with a negative sign, that's the coefficient on y2. So the eigenvalues and eigenvectors of that gamma matrix make it much more easy to interpret. If I just simply tell you the diagonal elements, that's not sufficient. But the eigenvalues give you those directions, those combinations, those y's, and then the eigenvectors tell you, is there curvature there or not? So you go back to that first example. Here's that gamma matrix where I have the off diagonal is a quarter, and here's I have the off diagonal, that's the square root of 2 that was chosen to give this a determinant of 0. So so for this matrix, there's the first eigenvector, there's the second eigenvector. So the combination of 0.23 times trait 1 plus 0.97 times trait 2, that's y1. y2 is 0.937 times trait 1 minus 2.3 times trait 2. It's just a new composite trait. And there are the corresponding, so in this direction there's the curvature, in that direction there's the curvature. What about the second gamma matrix? We'll call it gamma 2 for the second one. where now the off-diagonal element is here. Here is the first combination, so y1 is 0.58 times trait 1 plus 8.2 times trait 2. That direction has a slope of 0. The second y2 is 0.82 times trait 1 minus 5.8 times trait 2. That direction has a slope of minus 3. So these y's here, so in here, y is E1 transpose times z. It means that times trait 1 plus that times trait 2. y2 is E2 transpose times z. That times trait 1 plus that times trait 2. And so the nice thing about that is when you write them on this scale, all those interaction terms go away, and these things become far easier to interpret. And this is called the canonical A and the canonical B forms. And that just basically shows you that. And so, so these are called axes. These eigenvalues are called axes of symmetry, or canonical axes, or major axes, or principal axes. All the same thing. But the idea is, if you've got a quadratic surface, it's hard to interpret simply by looking at the matrix. The eigenvalues make it really easy to interpret. The eigenvalues tell you, is there curvature in certain directions? The directions the curvature corresponds to are given by their corresponding eigenvectors. So matrices are objects that describe geometry, and eigenvalues and eigenvectors are a way to extract that geometry from the matrix. Let me just say one more thing, and then give you a couple more things. We'll open for questions. So a really important question to ask is, well, are these axes meaningful? And the answer is they're quite meaningful. So let's go back and look at this example here. So if you have traits, very often people will do principal components, PCA analysis, right? Remember PCA analysis? Most of you have seen that. And in PCA analysis, I have a whole bunch of stuff. And I think, let's imagine I've got a whole bunch of environmental variables. And I may think that they're highly correlated, so maybe there's some environmental index I can extract out by weighting all those variables that will account for most of the variation. And maybe two such indices that are at right angles to each other that account for almost all the variation. Well, that's PC1 and PC2. And those people that have used PCs, for example, look at morphological traits, it's usually the case that PC1 corresponds to general measures of size. So all the coefficients tend to be positive, which means if any of the traits increase, you get that. So PCs are often hard to interpret. So, for example, PC2 are thought of measures of shape, but it's some weird combination. So people are a bit concerned about when you take morphological data and replace it by PCs, or take any trait data and replace it by PCs, some of the biological interpretation becomes less clear. Because traits are pretty obvious, you know what they are. When you create arbitrary combinations through the PCs, you don't have combinations of traits, and those can be hard to interpret. So because of that, people ask the question is, well, are traits now based upon canonical axes meaningful? And the answer is yes, because this isn't, if I do PCs, what I do is I take the phenotypic variance covariance matrix and I use that to create new combinations. We're not using the phenotypic variance covariance matrix. We're using gamma. Gamma is the matrix of how selection operates. So the canonical axes basically say this. This is how selection views those traits you're looking at. It says, in this combination, there's no selection. In that combination, there's stabilizing selection. Nature doesn't care that trait 1 is size and trait 2 is nose length. Nature says, for those traits, here's one axis of selection, here's a second axis of selection. So unlike principal components on a trait, where you're basically trying to move it around to maximize variation, and biologically it's kind of hard to say what that means, here, the matrix we're using is based upon the pattern of selection. So it's incredibly meaningful. So what it basically says here is that for these traits, it says, here are the axes selection acts upon. It acts in this direction with a certain strength. It acts at right angles in that direction with another strength. So I've taken selection and broken into independent axes where selection acts, and that's given by the gamma matrix, and therefore they're actually quite meaningful, right? So Mark Blows had a paper in Journal of Theoretical Biology about 10 years ago, and the nice thing about those review papers is they have the kind of paper, then they have, short page snippets by angry people or people with strong opinions, saying that they love this or this is complete crap. And then the author gets to then sort of, thank Professor Blowhard for their wonderful comments, but they're wrong. So they're always fun to read because it's a nice give and take. It's like being in a meeting where people are candid and open, they have a frank exchange. It's, oftentimes professional, but it's even more fun if it's not, let's be honest. And so the nice thing about this is one big issue people thought was, are these meaningful? And Connor said, the advantages are usually outweighed by the disadvantages. Results are not very biologically interpretable. Well, that's true for PCs, but biology doesn't care about how you define traits. Biology cares about what selection views those traits as. This is how selection views those traits. You may think your traits are natural traits, but evolution thinks differently. So the gamma matrix is nice because those canonical axes basically give you ways to think about how selection, how evolution views those traits. And in that sense, they're incredibly meaningful. So for example, it could be, here are your two traits. When you look at that trait, you look at the projection of how the fitness surface operates on that trait. So let's look at here. Here's trait 1. If you look at it, trace that curve. See that curve? That looks like stabilizing selection on trait 1. Look at trait 2, trace that inner curve. Again, it looks like stabilizing selection. But in reality, this is the nature of the fitness surface. When you project this complicated fitness surface on the two traits, you see evidence of stabilizing selection. But in reality, nature is actually quite different. So the canonical axes based upon gamma are indeed biologically quite relevant and quite meaningful, because nature doesn't care how you define your traits. This is how nature views the world. And so in that case, it's really quite meaningful. Let me give you one more example and open up for questions, because this is a key point. So Mark Blows has done a lot of stuff here. I love the little dinosaur, who Rob said, and Rob Brooks. So University of Queensland, I think Rob's at University of New South Wales or Sydney. I always get it mixed up. Anyhow, what they had noticed was, if you look at the gamma II values in 19 studies, that the eigenvalues is usually more extreme than the gamma II values. Let's go back to here. So if you look at the gamma I, you'd see 2 and 1. The eigenvalues say the real story is much stronger selection in one direction and no selection in the other. So these values here, diagonal values, are very misleading about the true nature of selection. And they said that because of that, you often get overestimates. That is, you take the magnitude of the largest gamma II, that's always less than the magnitude of the largest eigenvalue. So taking gamma II values is very misleading about the strengths of selection. And Lisa Kook, I put her up here because she and her group has done a lot of work of evolution in natural populations. They dug up and found the ever popular, well-known, I use that sarcastic, Mercer-Mercer theorem. That's like the Duran-Duran theorem. The Mercer-Mercer theorem says the eigenvalues, the magnitude of the largest eigenvalue is at least as great as the magnitude of the largest diagonal element. So it's algebraically inevitable that one trait combination will show stronger quadratic selection than any of the diagonal elements, provided they're not all zero. And here's an example. This is from, so Rob Brooks was a postdoc under John Endler. So here's Brooks and Endler looking at pigmentation traits in guppies. And here's the matrix they looked at, the resulting matrix for color traits. If you look at the diagonal elements, let's not worry what the traits are. What you see is, okay, modest disruptive selection and very weak stabilizing selection. That's what the diagonal elements suggest, right? So positive, positive, positive. So all positive curvature, disruptive selection. Negative, negative curvature. Again, as you use stabilizing selection, I mean, you've got a negative curvature. Let's look at the eigenvalues. There are the eigenvalues. 0.26. So there's one direction where there is very strong disruptive selection, far stronger, 4 times stronger than you'd suggest from any of these diagonal elements. Likewise, there's another direction where there is strong negative selection. Again, 5 times larger than anything you see here. So the diagonal elements here are quite misleading. By the way, your fun fact, this diagonal element, if you sum those elements, it's called the trace. If you sum those elements and sum the eigenvalues are the same. The sum of the trace is the sum of the eigenvalues. Random, interesting fact. So if you have a gamma matrix like this, the papers that report, remember those gamma values I plotted, in the King solver data? Those are misleading because they plotted those values there when in fact you want the eigenvalues of that matrix. And so here, if you look at this, you see 3 traits that show disruptive selection, 1 somewhat strong, and you show 1 trait that shows very weak stabilizing selection. If you actually look at what's going on, you have strong disruptive on 1 trait, weak disruptive, weak stabilizing, strong stabilizing. So using those values would give you a very misleading picture of what's going on. So the eigenvalues of the gamma matrix are nice because they give you a much better feature of the geometry. This is a deep property about eigenvalues in general. Eigenvalues are your best friend in multivariate data because they give you a much better feel for what the structure of that data is. If I simply have a list of eigenvalues, I can very quickly tell you a lot about the structure of the data, which I can't do by looking at that big matrix, which is really hard to interpret. Okay. So questions about gamma matrices and stuff? We'll start in the back and move forward. Sure. So what I haven't talked about is how you do statistics on eigenvalues. Turns out it's not trivial. There are ways to do it. There are bootstrap ways you can do it. There are also certain theorems, like there's something called the Tracy-Wilton theorem that tells what the distribution is. Unfortunately, I discussed most of that in volume 3. If you're interested, email me. I'll send you the chapter that talks about how you can do statistics on eigenvalues. Well, there's a method called double regression that people use, and that's one method. I'm trying to drill down the details. But the point is, there are methods that allow you to do statistics on eigenvalues, and they're not trivial, but a lot of them have to do with appropriate resampling. If you resample from a null, you can basically construct a null distribution. That's the basic idea behind it. Yeah. Four. So it's often called the rule of 4. Some people call it the rule of 3. If I'm estimating a linear thing like a mean, to have the same power, I need a sample size of roughly 4 times as large estimated variance. There's a great book called Statistical Rules of Thumb, and a rule of thumb means it's wrong, but it's close. By the way, people know where rule of thumb comes from? Don't get mad at me. This is history. So in England, it was legal to beat your wife as long as the stick wasn't thicker than your thumb. So some of these terms have really interesting history. Or what about, you heard the expression, the full 9 yards? It's like, why not 10 yards? Like, 9, that's length of machine gun belt was 27 feet. The whole 9 yards was in World War II. You shot the whole belt off. That's where these weird expressions come from. But the bottom line, though, is that you do need larger sample sizes. And the Walsh's rule of quantitative genetics is very simple. Your sample size is too small. Period. I mean, for example, Mark's lab, the Blows lab, I think does some of the best work in this. And their sample sizes are enormous. You know, they have minions chained to desks and stuff. And Mark's former student is gone now, so I can't. I'm sure she was chained to the desk while working on that. So you need big sample sizes. And so there's always issues of how uncertain these things are. And there are methods so you can put confidence limits on those eigenvalues. A deeper question is, how many eigenvalues are significant? Because it tells you how many spaces you have. And that turns out to be a non-trivial issue. And if you're interested, email me and I'll give you the stuff that we know about that. Yes? Mm-hmm. Yeah, so that's a great point because, again, I always recommend use different, analyze your data several different ways. And a good example of that would be Butch's example here. Oops, I went by it, didn't I? Nope, that's not it. No, that's not. I must have showed it last time. So Butch's example about, that's right, about reversal rates and stripe index. That's an example where all the power is in that correlation. And so by a canonical analysis might miss that, whereas if you simply plot that. So you want to analyze data several different ways because you can get different insight. The nice thing about a canonical analysis is it lets you get a quick visualization of what the fitness surface looks like. What you don't know is how it's rotated, right? The eigenvalues tell you the rotation, but the canonical axis will say in one direction I've got stabilizing, the other I've got disruptive. Then the eigenvalues tell you how they're rotated, so how that projects onto your actual traits. So you have the curvature and the rotation. And the correlation selection is just simply how the, what the rotation is like. Yeah, they basically give you, they give you the thing there. So there's so basically correlational selection, think of that as the tilt of that surface, right? And the tilt is relative to what you've defined as characters, how well do those match the actual axes of selection? And those angles are given by the signs on the eigenvectors. The eigenvalues tell you the general curvature pattern, and then how that curvature pattern is rotated with respect to your characters. Imagine like one of those circular plates at dinner that you move things around on. You have a curved surface on that. That curved surface is defined by the two eigenvalues. And then how you rotate that platter to match up with your traits, that's where the eigenvectors come in, and that's where you either have correlational selection or not. If it turns out that your curve axes exactly match up with your traits, there's no correlational selection. If you rotate it so the curve axes don't match up with your traits, then there's correlational selection. The relative importance of that, is more a matter of defining characters. I think the more critical issue is what's the curvature look like. Now, if the way you define characters have biological meaningful features, then the fact they don't match up directly with that curved surface may tell you something interesting biologically. It is symmetric because if you think about the terms in it, hang on a second. Let me just double check that. Let's go back to the books and books. Yeah, so it is symmetric because gamma ij and gamma ji are the same thing. Well, so the gamma matrix here is simply fitting the quadratic surface. If you have multivariate normality of traits, then their partial derivatives and mixed partials are equal. That's where that comes in. I mean, under continuity assumptions, and the multivariate normality gives you nice continuity assumptions, so you're good. That's the problems you run into with quadratic fitness surfaces, right? It's the simplest surface with curvature, and as you see, they're hard to interpret, but we've also seen how misleading quadratic surfaces can be, right? If you've got two fitness peaks, quadratic surface won't do it. In fact, oftentimes it will force a valley between your two peaks to try to capture that to give you very different fitness surface. So how do you handle those? And again, the notion is what we call semi-parametric fitness estimation. It's non-parametric in the sense that we're not assuming any functional forms. Like if I assume quadratic, that's parametric. If I'm not assuming any functional forms, it's non-parametric, but we call it semi-parametric because to solve these, I have to make assumptions about how the residuals behave. Yeah, blah, 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 blah. So one approach that people use is something, and Schluter used this to go from one trait to multiple traits, is this pursuit projection regression, or PPR. And the idea behind this is two things. Number one, there's an optimization routine you go through that weights your residuals. So if you have Poisson versus binomial residuals, you weight them differently. And then that generates an optimal axis of variation. That's your pursuit vector. Then a second at right angles, then a third. Once you get your pursuit vectors, so here's the projection vectors, AI, and they have associated ridge functions. Those ridge functions are the things that are non-parametric. Those are splines. So I have a direction, and in that direction, maybe I have a double hump. That direction is given by the projection vector. That double hump is given by the ridge function. And think about those ridge functions as each of those is being fitted by a cubic spline. So Schluter had the method to non-parametrically fit in one direction. Well, you basically get the direction by the projection, and then the ridge function fits them. So I end up getting these things here, where xi is this axis based upon projection vectors. Those projection vectors don't necessarily correspond to the eigenvalues of gamma, because you're optimizing something differently. Gamma is finding the best quadratic fit. The pursuit projection is trying to optimize it without making any assumptions. So it uses a criteria to get the projection vectors and then fits those. So we'll give an example in a second where these don't necessarily match up with that. And so one way to fit, another way to fit data is if I have a set of axes I've defined, I can use what are called Thinplate splines. So a spline is a quadratic that joins adjacent quadratics together. Thinplate splines are that idea, basically thinking about create a wireframe and put little plates on that wireframe. And so I can basically build up a non-quadratic structure like that. So let's look at an interesting example. So this is a Thinplate spline fit, and these are pupfish. And if you take the fitness data, which is based upon survivability on certain traits and stuff in vernal ponds or following, if you take the fitness data and fit a quadratic to it, you basically get one hump. If you do Thinplate spline fit to it, you get multiple peaks, and the multiple peaks correspond to where current populations are sitting. So if you simply fit a quadratic here, you'd miss the multiple peaks and you get an assumption that's quite different, whereas fitting the Thinplate splines gives you one that basically allows you to capture more of the biological conclusions. So the key part there isn't this example, it's that when you have a quadratic surface, you are forcing at most a single peak or a single valley or a single point about which peaks and valleys emerge from, a single saddle point. When you have a quadratic surface, there's one extremum point, and that's it. If your data is naturally two or multi-humped, then you want to use Thinplate splines or pursuit rejection. So here is an example from Blows. I think Mark was involved, I think Rob Brooks was involved in this as well, on, again, traits in guppies. So spot size, spot placement, color length, and females use these to make choice of males. It's like, wow, check out those double spots. So when you go ahead and take those data, here are what happens if you fit a quadratic. So here's the first eigenvector. Here's the second eigenvector. Conversely, if you use pursuit regression, you can look at the first projection vector. Here it is. And the second projection vector. You can then look at it's easy to compute the angle between two vectors. So the angle between E1 and A1 is 88%. These things are almost at right angles to each other. So when you get your axes based upon gamma, you can get those axes and then put Thinplate splines on them. You often get very different axes than if you get your axes using pursuit regression. So these two are the axes of variation if you assume the curve is quadratic. These two are the axes of variation where you don't make that assumption. And what you basically end up getting, and by the angle between the E2 and A2, so the second eigenvalue for gamma and the second projection vector is again almost 90%. Here's what the data look like. So if you simply fit a quadratic, you get disruptive selection and all with it going off in either direction. Again, y1 and y2 are these combinations. So y1 would be this times trait 1 plus that times trait 2 plus that times trait 3, etc. y2 would be that times trait 1 plus that times trait 2 plus that times trait 3. X correspond to weights using projection. So trait 1 times that plus trait 2 times that, etc. is x1. Trait 1 times that plus trait 2 times that, etc. is x2. So if I use these two combinations, y1 and y2, there's the surface I get. If I then take those axes, y1 and y2, and fit thin plate splines, I get this structure here. It's a bit different, right? For example, it goes down here, but it's using these two axes to capture that. If instead I take as my canonical axes A1 and A2, so now my new combination is this times that plus that, trait 1 times that plus trait 2 times that. That gives me x1, trait 1 times that plus trait 2 times that, etc. gives me x2. If I look at that combination, here's what I get. So here is thin plate splines based upon using the axes from the quadratic regression. Here is projection regression using the new axes. And as you see, there's some similarities about these surfaces, but there are also some significant differences between them. So the non-parametric fitting is somewhat of an unresolved issue, and the main problem you have is you're limited by how dense your data is in terms of scanning that surface. If your data is very loose with regard to that surface, you won't pick up a lot from it. If your data is highly packed regarding that surface, you can pick up a lot from it. So that's what I wanted to say about multivariate geometries. One final point. Remember, when the geometry is well behaved or the data is multivariate normal, we have that the beta equals the average value of the individual gradient. What happens if I don't have multivariate normality? We could use the Jansen-Stern gradient. By the way, it was nicely pointed out that I have a typo probably all throughout the book. That should be Stern, not Stein. I got carried away thinking about the James Stein estimator. So when you're dyslexic, it's fun. There's all sorts of errors that you read through and they read perfectly normal. So their way of computing this beta, remember, so if Z is multivariate normal, if I simply do a linear regression of fitness on traits, I get beta back out again. Beta equals this. If data is not multivariate normal, if I wanted to find my gradient as the expected curvature of the individual fitness surface, then I can compute these things numerically. And I can do that for these non-parametric estimates. Likewise, if Z is multivariate normal, I have this nice feature where the beta, that is the best linear term, corresponds to gradient with respect to individual fitness surface. But if I don't have multivariate normality, again, I can use a Morrissey method and calculate this value numerically. So under multivariate normality, the best slope of a linear regression, the average curvature of the individual fitness surface, and the gradient of the individual fitness surface with respect to means are all the same. If I go away from multivariate normality, beta is still, by definition, the coefficients in the best linear fit, but to get this gradient based upon average slope, I have to solve it numerically. If I want to get this gradient based upon landscape gradient, I have to solve that numerically. This is the relevant one because this tells us how the population is going to change. A couple more quick things and we'll see if there's questions. So fitness surfaces can change over time. So here are two surfaces from this lovely plant nearby, in the Rubaceae. And what you see basically are, these are in what times? June of 1985 and July of 1985. And what you see is 4 traits, stem diameter, internode length. The fitness surface has changed rather dramatically. Now, the big asterisk on these is you look at that and go, wow, those are so different. What you don't know is the confidence interval, right? So data are often measured with some precision. So even though these look dramatically different, these in theory could be very similar just due to sampling noise. But the bottom line is fitness surfaces can change over time, and that leaves you with a quandary. Do you estimate them individually, therefore reducing your power, or do you combine the estimate, potentially obscuring things? And there's no right answer, except the right answer is try them both and see what the data suggests to you. So fitness surfaces can change over time. They can also change over space. There's our old friend, the great hit again. And this basically is fitness measured by fecundity and two composite traits, M1 and M3. Here is a location. Here's a location just 4 kilometers away. So again, these surfaces can change over time and over space. Some of these differences are simply estimation noise, because it takes a lot of data to estimate these well. Others are real biological differences in terms of temporal changes or spatial changes. And the issue you always face is, do I combine these estimates or estimate them separately? And I would recommend you do both and look at for consistencies. Okay, so that takes us basically through fitness surfaces. We talked about geometry of quadratic fitness surfaces. We talked about non-parametric methods to estimate those fitness surfaces. So questions about this? So I want to end up by talking about path analysis. Path analysis is essentially a model of assumed causality. So if that model of causality is good, you have a lot more inference. If that model of causality is bad, you've got a lot lower influence. So I want to talk about path analysis assumes a causality structure. I want to talk about if I have a path, and basically trait 1 affects trait 2 and trait 2 affects fitness, that sort of thing. I'll give examples. I'll talk about selection analysis on the path diagram. We'll talk about Morrissey's extended selection gradient vector, selection on latent variables, and elastic path diagrams. So here's an example of how a path diagram would work, a very simple example. Suppose, so in a path diagram, single arrows imply assumed causality. So weight affects height. Double-headed arrows say we don't know these traits are correlated, I'm not sure how. So in this example here, and by these path coefficients turn out to be correlations. So the correlation between weight and height is 0.35, and the correlation between height and fitness is 0.15. So if I didn't know height, what I would observe is a correlation between weight and fitness is that times that, because you can multiply correlation coefficients to get the correlation between weight and fitness. So for example, in this case, I'd basically get about 5%. So that times that says the correlation between weight and fitness is 5%. If I have this causality structure and I can measure the associations, what I see here is that weight has no effect at all on fitness. Rather, weight influences height, and height influences fitness. So the path diagram here gives you this causality structure. Weight affects height affects fitness. If you assume that causality structure, you'll get a better inference about the nature of selection. Selection isn't on weight, selection is on height, and height counts for about a 0.5 squared. Remember, correlation squared is percent variation accounted for. 0.5 squared of the variation in fitness. So path analysis assume causality. I'll give examples in the next slide. And so the result is they're extremely dependent on the assumed causality structure. So as Kingsolver and Schlemsky said, uncritical application of path analysis, the analysis selection, is likely to lead to misleading and erroneous results. Conversely, if you have a right path, and path typically means I look at how I assume characters influence each other, then from that, that's my assumed causality structure, then I estimate how those individual characters affect fitness. If that causality structure is pretty good, then you can actually get a lot more inference. So here are 3 examples of paths. So I have, what do we have here? I have, so I have seed weight S, plant height H, leaf area A, and number of leaves N. So there they are. There are my 4 characters, and they potentially affect fitness. This is what Landy Arnold assumes. Landy Arnold assumes the 4 characters are correlated, and they all potentially increase fitness. So you estimate the correlation, estimate the fitness. Let's still imagine that seed diameter affects leaf area, leaf height, and number of nodes. And seed directly affects fitness. So then once you have this diagram in, you can then ask, okay, is the pathway from here to here, that is the direct pathway, significant? Is this pathway significant? Is that pathway significant? So this here is a correlation structure, and the double-headed arrows here, I make no assumption about how one trait influences the other. Here I do. I said seed weight here influences these 4 traits. Or it could be that seed weight here directly influences fitness, but then it influences height. And height then influences leaf area and nodal length. So these different causality structures give different ways that selection operates on you. And so under Landy-Arnold, you're assuming this. Different causality structures then give you different pathways for how selection can operate. So if you feel relatively comfortable, and the causality structure here is simply draw the arrows connecting. Once you draw the arrows connecting, you then estimate the weights. So causality structure isn't the arrows and the weights, it's the arrows. Then you go back and estimate the weights. So causality structure is this graph about how one trait influences other traits. So what about regression versus path analysis? So basically, if this is the observed order, germination leads to growth, leads to flowering time. Regression simply uses the correlations among these 3 things, whereas path analysis explicitly uses this information. So here's an example from Tom Mitchell-Ols's group. And so what you're looking at is germination date, seed weight, June size, elongation weight, early growth, late growth, and fitness. And what I've shown here are only paths that are significant or shown. So once you assume this causality structure down here for the traits, you can then go ahead and estimate these path coefficients. You can then go back then and estimate path coefficients in here. So I think in the example I talk about the path from here to here, you can actually estimate it and give a number, but it's not significant. So this is the assumed structure. And in this assumed structure, if you then go and look on this structure, and again, this is basically regression analysis once you put the path down, you see that the correlation between germination date and fitness is minus 0.08. So earlier germination leads to higher fitness. You see that early growth and late growth both affect fitness. So now you can look at the different paths that germination date affects fitness. Directly, indirectly, through germ size, early growth, and then on to fitness. Indirectly through germ size, early growth, and how early growth affects late growth, and then on to fitness. So I've got direct paths, and I've got indirect paths. So the nice thing about a path analysis is it gives you a much deeper understanding for how selection may operate. So little e's basically are residual errors that come in. So in your regression model, I can predict certain things, then I've got additional noise. What this basically means is I have no idea where this comes from. So early growth is influenced by this pathway here, plus other stuff. And that other stuff is assumed to be independent. There's no arrows connecting them. So this is basically residual noise. So this is the residuals in a regression. Same thing up here. So the fitness up here, these are causal factors, and this is everything else. So oftentimes these are much larger in terms of how they explain than the causal factors. But the idea is they're independent, they're just random noises added to the system. So you could, well, in the sense, you could have, for example, suppose you have soil type. You could measure that, so you'd have germination date and soil type, and maybe soil type influences fitness as well. So if I can measure environmental factor, I can put it in the causality diagram. If it's unmeasured stuff, those aren't in the causality diagrams. So I could certainly put in measured environmental factors if I want, if I know what they are, and then I can ask how well they fit. So remember, the key idea is in a path analysis, I basically write down a causality structure with potential arrows. I then go back and see which arrows are significant, leave the ones that are, that gives me my causality structure. Then with that causality structure, then I go ahead and look and see how each of these things affect fitness. So for example, there's an arrow from here to here, and even though I get a value, it turns out to be non-significant. So in a path diagram, usually you only display significant connections. The numbers here is a correlation. The other approach you often see is thickness of the line sometimes is used to represent strength of correlations as well. That's the path diagram. So single arrows, so late growth influences fitness. Double arrows means I simply have two traits which are correlated. I'm making no assumption at all about how these traits are connected. Path analysis, by the way, traces back to Sewell Wright. Remember Sewell Wright, Wright-Fisher model? So he's the one that put this together. I mean, not the analysis for selection, but path analysis in general. Yes, it assumes linear relationships. You can account for nonlinear relationships by putting in dummy variables that basically talk about quadratic effects. And the notes give you, the book gives you a link to a couple of papers that talk about that. So right now, and this has all the issues we've talked about in that we're assuming that these residuals here are all normal, right? So the issues about normal residual fitnesses we talked about before, they're also a problem in path analysis because this is simply, we turn this into a regression model, and these are simply assuming these residuals are independent and normal. Mm-hmm. And we'll see that when we talk about Morrissey's, because that's what Morrissey basically looks at. Yeah. So that's an example of a path analysis. And once you put the path down, then you simply estimate those strengths of those traits on fitnesses. Right? So here's an example from this. So if you look at seed weight, there's seed weight there. It turns out that there's actually a weak path that's not included in the analysis, in the model. Why? It's not significant. But so seed weight has a path that's about 4%, but it also has a path which is that times that. That's the path getting you there, and a path which is basically that times that times that, which is getting you there. So I've got one direct path here to there, and then I've got these indirect paths. And so if you look at the indirect effects of fitness on seed weight, it's 0.1. If you look at germination date, I've got a path down here of minus 0.08. Then I've got paths through there, so that times that times that, a path through there, that times that times that times that. And it turns out the indirect effect is given by here. So likewise, you can go through. So for example, here are the direct effects. So late growth rate, that's a good example. Late growth rate down here, there's nothing, no other trait late growth rate influences. So late growth rate has a direct effect on fitness and nothing else. Early growth rate, direct effect on fitness and indirect effect through these two paths. So the paths allow you to look at the direct effects and the indirect effects through different characters. Yeah. So I guess one thing you could do is you could perturb it and look at how stable the R squareds are in terms of not throw fitness out and look at this and actually ask when you change those causalities what happens. Or if I have a different topology. So the key here is you're making a topology about what trait affects what. You could change that topology and look at how stable it is. If you're worried about maybe several causality structures being correct, put them all and analyze them all separately. Think of this as an exploratory tool, not something that's going to give you exact p-values. And the exploratory tool lets you look at, if this is the causality structure, what traits have the most direct and indirect effects? If I don't assume that causality structure, what do I have? Right? Yes. Yeah. Yeah. So again, it's one tool, and as any tool, it has strengths and weaknesses. The nice thing is it can allow you to have a better interpretation of your data, right? So Michael Morrissey had an issue called the extended selection gradient. And look at these two situations here. So suppose that F is a character I don't measure. Path 1 and path 2 both generate a correlation between Z and W. The fact that I left out F in path 2 is no big deal. The path that I left out F here, that's the missing trait problem, because Z is not directly causal on W, whereas here Z is indirectly causal. Here, the correlation is not caused by causality, it's caused by something else. So those are traits we want to look at the difference, because here we don't care if we don't have F. Here, not having F gives a very different interpretation, because Z never affects W. Here, Z does affect W, but indirectly. So Morrissey came up with this concept of extended selection gradients, eta, and what it does is it measures the total selection caused by a trait and its downstream causal components. And the idea here is that if I leave out a trait, eta doesn't change. So down here, eta would have a value of 0, but here, eta would have the same value if I included F. Eta is either the same value if I included F or didn't include F. So the motivation is, here's the within generation change. We've seen that before. And so let's let beta, path analysis I, denote the selection gradient from the direct effect of a trait on fitness. There's the path, the direct path from I to fitness. Likewise, let capital phi of ij be the total impact of all connecting paths of the effect of I on trait j. We'll look at an example. So then by analogy with here, since your correlation with yourself is 1, the extended gradient on trait I is the direct effect of I plus the indirect effect where I influences trait j and direct effects of trait j. So that's his notion of extended gradient. He then showed how you can compute this matrix here, phi, and you can get phi by taking the product of all paths connecting I and j and summing over all those paths. But Michael had a quicker way. He said, suppose I have a matrix B of path coefficients, and the matrix are constructed the following way. If I directly influences j, not through an intermediate, but directly, then Bij is the path coefficient between them. If I does not directly influence j, so I influences k, influences j, I does not directly influence j, we give it a value of 0. So I'm only counting here paths that directly connect it. There's no intermediate step. If I then use that B matrix, then Morrissey found that phi equals the identity matrix minus B inverse, and that gives you the extended selection gradients. So in terms of path 2, Z to F to W, the value of NZ equals the same value in a model where F is ignored. So the nice thing about these extended gradients, if I've got direct effects on fitness, if I ignore intermediate traits, I still recover the same value. So here's an example. This is an example from number of spikelets in wild oats is the measure of fitness. And so what I have is I have date of germination. Let's see, M60. Yeah, I've got date of germination, mass at day 60, days to fruit flowers, total mass, number of tillers, and mass of tillers. So date of germination affects mass at day 60. That affects total mass, and it affects days to first flower. So total mass affects mass at 60, affects days to first flower, and total mass, days to first flower, affects reproductive tillers and mass of reproductive tillers, and mass, total mass also affects fitness. So there's the causality structure, and the idea is these three components, mass, number of reproductive tillers, mass of reproductive tillers, those are the three traits that directly affect fitness, and then here is the other pathway. And again, the idea is the topology is given by here, we ignore fitness, then given that topology, we go ahead and test these. And I've labeled these 1, 2, 3, 4, 5, and 6 to give me the 6 traits. So that's my path diagram. And again, what you see is that these traits here only affect fitness indirectly. whereas these three traits here directly affect fitness. So here is a gradient. So germination, 60, days of first flowering, mass, reproductive tillers, reproductive mass. If I did Landy-Arnold, those are the weights I would get using Landy-Arnold. Landy-Arnold completely ignores that causality structure and just runs it as a regression. However, if I actually go ahead and look at the path analysis, the only direct paths are mass to W, reproductive tillers to W, mass reproductive tillers to W, and they're the values I get for those three traits. So you see you get a very different interpretation on those. What are the extended gradients? Well, here's that B matrix. So let's look at it here. Trait 1 only directly affects trait 2. Trait 2 only directly affects traits 3 and 4. Trait 3 affects 5 and 6. And trait 4 affects trait 6. So if you look at that, those are basically the only single-step connections. That's your B matrix. So for example, over here, it says that trait 2 affects traits 3. Sorry, it says that traits 2 affects 3 and 4. So there is 2 affecting 3 and 4, and there are the coefficients on it. That's your B. If you compute this matrix of interaction here, you get this. And to see it's correct, we can actually look at something. So for example, this expression here asks about the effects of date of germination on trait M60. And so you can kind of go, so that's a direct path. A more complicated path is trait 1 on trait 5. So trait 1 on trait 5 can go 1 to 2 to 4 to 5, or 1 to 2 to 3 to 5. So, dollar sign's interesting, missed that. So if you multiply these coefficients here, you get that. Multiply those coefficients there, you get that. So that's how they come in. You then go ahead and compute the extended path coefficients. And basically, here they are. So it's theta times the direct path coefficients. These two don't change. Why? Because there's nothing downstream. These go there directly. Whereas mass, so how come mass is different? Because these only affect fitness. Mass affects fitness and it affects these traits. So there's a direct effect, then there's an indirect effect through here. So resulting extended gradients basically become this. So if I leave out any of these intermediate traits in my analysis, these results here aren't changed. So that's the notion of an extended gradient. And the idea basically is to account for the fact that you can influence downstream things. If we don't include those downstream things, you still want the gradients to remain unchanged, and it would in this analysis. Yeah. So elements of B basically are given by the path. So elements of B basically say, which traits does trait 1 impact? It only impacts trait 2. What traits does trait 2 impact? It only impacts 3 and 4. Those are from the path analysis. You do the path analysis and you estimate, these are correlations. It says a correlation between mass at day 60 and date of fruit flowering is minus 0.2. So these are standard path analysis coefficients. Path analysis is a partial regression. I standardize the variables and do a regression. That's exactly where it comes from. So that's Morrissey's extended gradient. So another application of path analysis is latent variables. Suppose that, what's a way to try to get around these missing character problems? Suppose I've got several things I measure, and all those are correlated with the trait, where the trait is correlated with fitness. But the things I measure are not correlated with fitness. A good idea is I measure several morphological characters. They're all correlated with size, and size is what determines fitness. So here's the idea. In a Landy-Arnold model, I'm assuming correlations between these traits that influence fitness. In a latent factor model, I imagine I've got size. Size affects fitness. It affects these two traits, and these two traits may affect fitness. So I can actually use information from here, for example, maybe a PC1, and make a new latent variable and put that into the model and run it, and then ask, when I put this factor in the model, how much of these correlations remain. I'm kind of giving you a cartoon version. I won't go into the details. That's an idea where you can basically, by putting in a surrogate variable, you can basically ask, does this latent unmeasured variable have some sort of impact? The kind of last use of path analysis gets back this notion of elasticity. Elasticity says, if I have a life history component and I make a proportional change on that, what's the proportional change on overall fitness? So it's taking life history components and trying to map those changes into overall fitness. The second aspect is, if I have a trait, how does a proportional change in that trait result in a proportional change in life history component? So I can then want to make the connection between a proportional change in a trait and how that maps into fitness. Elasticities of trait on life history times elasticity of life history onto overall fitness allows me to make that connection. And recall that those elasticities for trait values on life history components are given by the mean standardized gradients. So here's basically the idea. I want to ask how change in a particular trait influences the overall growth rate. Well, that's basically summing up over all the fitness component it impacts. Here's the change of how fitness component I changes the overall growth rate, and here's how a change in that trait changes fitness component I. This is the elasticity of fitness component I on the overall growth rate. This is the elasticity of our focal trait on fitness component I. So by summing up over those, I can ask for the total impact, and that'll basically tell me which parts of life history, which traits most impact that. And here's an example from salmon. So I basically have two traits, growth and early entry into the stream. I then have life history components, newborn survival, juvenile survival, migration survival, fecundity. So this is the selection analysis. I can then go ahead and compute variance standardized gradients on how growth impacts these traits. And what you see is it's only significant on these three. It doesn't influence migration survival. Likewise, entry date only influences migration survival. So likewise, I hate these things here. Likewise, I can ask how newborn survival, how does it affect growth rate? What about juvenile survival? What about migration survival? What about fecundity? So here are the elasticities. So proportional change in newborn survival results in a 0.25 proportional change in growth rates. So elasticity diagrams basically allow you to connect that. So what is the effect of growth on the growth of the individual on the population growth rate? Well, it's simply that times that plus that times that plus that times that. That's the total impact of growth on population rate, and you can see the various components. What's the impact of entry date on growth rate? It's that times that. So you can go ahead and work that out there. So the impact of migration survival has an elasticity of 0.9, and migration survival affects lambda of 0.25. So the impact of migration survival on overall growth rate is 0.23. So a 10% increase in mean entry results in a 0.23% increase in growth rate. If you look at juvenile growth rate, it affects three traits. So it affects newborn survival. Newborn survival affects lambda. It affects juvenile survival. Juvenile survival affects lambda. It affects fecundity. Fecundity affects lambda. You add all those up, and the total impact is 0.72. So a 10% increase in growth rate results in a 7.3% increase in lambda. But that's mainly due to this component right here, its impact on fecundity, because its impact on these other two traits, juvenile survival and newborn survival, is actually negative. So these elasticity path analyses are nice because they allow you to put a whole bunch of stuff together and basically make inferences about which causal component affects which thing. So again, it's another nice tool. These path analysis tools are kind of nice, what I would call auxiliary analyses. When you get a certain analysis, you can try to refine it and learn some more basic biology by drilling down on these things and getting some more information. So yeah, this one here, yeah. Sure, you can certainly break this. You can certainly break this further, but well, the thing is, you can always break stuff further, and so what you're trying to do is capture the things you can easily measure. As you break things into more and more components, you lose power. So you have to ask yourself, are those reasonable things to measure that biologically make sense? Because you can always make models more complicated. But Einstein had a great line. He says, make the models as simple as possible, but no simpler. So those boxes determine a bunch of stuff. So if you're worried about components of fecundity, you could break that up, and then you'd have additional boxes there, fecundity opponent 1, fecundity opponent 2, and you can ask about what their effects are, their elasticities are on the growth rate, and you can ask what the elasticities are on lambda, and you can ask the elasticities of juvenile growth rate on those components of fecundity. Uh-huh. But remember, so that non-independence is taken care of in the elasticities. So elasticities take that Leslie matrix and ask if I change those components. So it doesn't matter if you get there. The elasticity has that taken into account. Or they could be representative stages. Your analysis may not have everything, but what it says is for those things you measure, here's what you have. So for example, you could imagine those as L1 and L2 off-diagonal elements of that matrix, and fecundity, let's say, is B2. You think about that matrix. Well, again, don't think about a one-to-one mapping. The idea is that you've got a Leslie matrix, and you've examined certain components of that matrix. Maybe not all components, because you can't maybe measure all components. But you've examined certain components, and you then ask, what are their elasticities on the growth rate, and also what are the elasticities of traits on those components. So think about it loosely. Don't drill down and worry about how you can make it more complicated. Think about the basic idea. If the basic idea makes sense, then you can extend that to more complicated situations. Don't worry about the complicated situations first, because they confuse things. Think about the basic idea. If the basic idea makes sense, then you can, on your own, figure out from that basic idea how you might extend this to more complicated settings. So the last thing I want to talk about, just a couple of slides, we'll actually finish a couple minutes early, is levels of selection. And we saw an example where, so Mike Wade, one of his students, found that larval group side had an impact on fitness of imported leaf beetles. Larval group size is not an individual trait. So if I'm measuring a Landy-Arnold fitness, I might measure the size of a larvae, how quickly it grows, but larval density or number of larvae around you is not an individual trait, so I wouldn't include it in my regression, and yet I'd be missing an important component of selection. So how you incorporate that is actually very straightforward, and it's got this fancy name called contextual analysis. The bottom line is you simply add additional traits. So I'm looking at you, I'm looking at your larval character, and then I also add on the group size you're in. I simply add that group feature, and that's a trait I assign to you. I would also assign that group feature to everyone in your group. So you can have aggregate characters. So it could be the mean weight of all the larvae, or you can have global or emergent characters, such as the density and things of that nature. So an example of that is this nice little plant in Finland. And in that plant in Finland, they're basically concerned with survivability. And so they measured plant height, Z1, number of stems, Z2. Then they measured average plant height in a group and number of stems in a group, because these things occur in group. Then they measured plant density. These are aggregate features, sum over individual values. That's a feature of the entire population density. So that's the model they fit in the regression. So for example, everyone in the same patch would have these values the same, but these values would differ. As you go across different patches, those values would differ. You then go ahead and run the regression. When they ran the regression, what they saw was height was significant, but so was mean height. So your fitness is equally determined by how tall you are and by the average height in your group. Mean fitness was influenced by stem number, but it was equally influenced in the opposite direction by the average stem number of the group you're in, and also was influenced by density. And they made the great suggestion, this is probably predation due to reindeer, right? That's kind of cool. So the whole idea about this simple example is you can add in group level traits into a standard fitness model and simply test if those coefficients are significant or not. And that's how you can extend and look for, is there selection operating above the level of the individual by putting in potential traits like mean, averages, or group level traits and ask if they're significant. And how you extend this and deal with selection is covered much more extensively in chapter 22. And that, I think, is your 5-day overview of all the methods to estimate selection. We started with phenotypic fossil data, data in messenger RNA expression, moved into markers, very sophisticated use of markers in genomic scans, then moved into ecology, measuring individual fitness, then moved into statistical issues about if I've got estimates of individual fitness and phenotypes, about assigning and connecting those. So that's kind of your broad overview. The notes give you details. And again, we covered a huge amount of stuff. What you should take away from this is an appreciation for different approaches if you hadn't seen them before, and maybe some rough idea about how things might happen. If you have that rough idea, and more importantly, have the appreciation, you can then go back and look in the notes or look in the book or look in the literature and actually get all the details you need to apply these things to your specific situation.